So I'm a formulation guy, so I definitely belong on the development end of drug discovery and development. I ditched inorganic chemistry, no I didn't, I kept inorganic. I ditched organic chemistry in second year because I couldn't really do it. I wasn't very good at synthesizing molecules and columns and things like that. So what I want to talk about this morning is how we can get the drug to the right part of the body once we've developed it. So we have our fantastic new medicine, whether it be a natural product or a protein or a synthetic drug molecule. How can we make sure that we deliver it to the right part of the body at the right time and in the right amount? Because all of those things are required in order to make a successful medicine. And I think Dr. De Silva will talk about this a little bit more after lunch. But first of all, I thought I would just tell you a little bit about UCL. So we are situated in London, unsurprisingly, given the name is University College London. And this here is where the Queen lives, and we're based just up here. So we're about 4 miles from Buckingham Palace, right in the heart of London. And UCL was founded in 1826, and it was founded on the basis that everyone had the right to a university education. Prior to that time in the United Kingdom, in order to go to university, you had to basically look like me. You had to be a white man, and you also had to be a Christian. If you didn't meet all of those criteria, then you couldn't go to university at the time. And UCL was founded on the basis that everyone, if they were smart enough, if they were clever enough, had the right to university education. And we were founded really based on the ideas of this guy. This is a chap called Jeremy Bentham. And he was one of the people who had these ideas about opening up university education to everyone. And he left us a lot of money in his will when he died, for which we're very grateful. But he was a little bit crazy, and one of the conditions of the will was that in order to get his money, we had to display his body in the university at all times. So this is not old, this is a contemporary photograph of his body. And if you go to this building, which is our main building, in this corner of the building there's a big wooden box, and in that box sits the mummified carcass of Jeremy Bentham wearing his original clothes. The head is made of wax because in the 19th century when he died they weren't very good at preserving things, and so the head, which you can see here between his legs, is rotting rather. So the head on his body is not real, but the body is real and the clothes are real. And when the most important committee in the university has its meetings, then what they do is they take the head along in a box, and in all of the minutes of the meetings, it says that Jeremy Bentham is recorded as being present but not voting, which I think is appropriate because he wouldn't make very good decisions. So what I want to talk to you about today is a technique called electrospinning and how we can use that to make polymer fibers in order to target a drug or drugs to a particular part of the body. So I like electrospinning because it's easy and it's cheap, and that means it makes excellent student products. So what we do in electrospinning is very simple. We take a volatile solvent, ethanol, methanol, chloroform, something like that, and I add to that a polymer, and also, for my purposes anyway, a drug or drugs. And what we do is we squirt that solution out of a syringe fitted with a metal needle tip. And I squirt it towards a metal collector, and between the two I apply a massive potential difference. So I'm applying a potential difference of maybe about 15-20 kilovolts, which sounds dangerous, but the current is remarkably low, so it's fine, and if you touch the needle then you don't die, although you do get a bit of a shock. And this is a really good approach because it's very fast. The whole process happens on about 10 to the minus 2 seconds, and you go from liquid to solid very quickly without requiring the use of heat. So there are many pharmaceutical formulation approaches which go from liquid to solid. It's a standard route, spray drying, hot melt extrusion and so on. But the problem with those approaches is they use heat. And many drugs are heat labile, and therefore if I can avoid using heat, that's potentially very beneficial. Heat is of course also expensive. So this is what the equipment looks like in our lab in UCL. Here's a power supply. Here's a syringe pump we can use to expel the polymer solution. Here's my collector. And you can probably just about see there, there's a crocodile clip, and we apply a potential difference, a voltage between the clip and the collector. So if I just have a solution, then when I squirt it out of the needle tip, what I'm going to get is a pendant droplet, just like this. But when I apply the voltage because electricity and liquids interact very effectively, what will happen is the surface of that pendant drop will become charged. And the charges will all be the same, they're like charges. And that means that they're going to repel each other, which causes, I don't know how easily you can see that, but I promise it's true, which causes the droplet to deform into a cone shape. So we get kind of this shape. And the charge is still building up on that cone. And so in order to dissipate the charge, what the cone will do is it will eject polymer jets. And that's what's happening here. Here's the cone, here's the polymer jet being ejected. And that polymer jet is ejected towards a grounded collector plate. So typically the needle is positive, the collector is grounded, and the jet travels towards the collector in order to try and discharge its charge. And what I get out are one-dimensional nanoscale fibers, which are woven together to form a map kind of like this. So here's a schematic, and here's a scanning electron microscope image. And the best way to think about this, for a Sri Lankan audience anyway, is to think about string hoppers. These are just like string hoppers. The individual strings you can see, and there are no connections between the strings, but they're interwoven. So if I want to take a string hopper and pull out a single string, that is a difficult thing to do, even though the different strings are not held together by any particular forces. So we have these things, and they're porous, they have junctions between them, one-dimensional nanoscale fibers. So I said the process is very fast, and that means typically what I get are fibers with a functional component like a drug, in red here, randomly distributed throughout that fiber. But if I want to, I can do a more sophisticated experiment, and I can use two needles, one needle on the outside and one on the inside, and I can use those to make core shell structures like this. So here we have a shell made of one polymer, a core made of a different polymer, and drug distributed in the core. We call this coaxial electrospinning, and we'll come back to it in a little while. Okay, so that's cool, but why do I care? Well, I care because if I change the polymer I use, then I have the opportunity to deliver a wide range of different drug delivery profiles. As we all know, I'm sure, most medicines are given orally, just because it's the most friendly way to take a medicine. It's likely to be the highest patient compliance. And most medicines that you take, most tablets, capsules, are what we call immediate release. That means that as soon as the medicine hits your stomach acid, it's going to start dissolving, start freeing the drug in solution. And I don't know if it's true in Sri Lanka, I guess it probably is, but our British Pharmacopoeia definition for immediate release is 80% of the drug loading is from the solid into solution inside 15 minutes. And so that can be fine. If I have a headache, I really want my aspirin to dissolve quickly so that I get therapeutic benefit. But we talked earlier about some of the challenges we face in developing new medicines. And one of those is that immediate release is not always what I want. Let's suppose, for instance, I'm giving medicine to a child. A young child is not going to understand why they don't feel better quickly. Also, they're not going to be able to easily swallow a standard-sized tablet or capsule. So it can be very desirable in such instances to make what we call a fast-dissolving formulation. Something where I can make a wafer, put it on my tongue, and it dissolves within a few milliseconds, freeing the drug into my mouth, which is highly vascularized, and so which can absorb the medicine very quickly. So we might want something that dissolves really quickly within a few seconds. I may want delayed release. There are a number of drugs which are rendered inactive by stomach acid or by enzymes in the stomach, or where I want delivery to the small intestine or large intestine because I get better therapeutic benefits. So we call these delayed release formulations, and the simplest example is an enteric-coated tablet or capsule. Just has a pH-sensitive coating, which means it can't dissolve in the stomach, but it can dissolve later on in the GI tract when the pH is high. We're getting cleverer. We might want extended release, maybe release over the period of a day, so that I only have to take one tablet in the morning rather than 3 or 4 throughout the day. I'm getting even funkier. Maybe we want pulsatile release. This is bursts of on-off release, which can be very useful to treat diseases which follow the body's natural circadian rhythms. Things like epilepsy and rheumatoid arthritis are worse at different times of day, and various other conditions are better or worse before or after release. So on-off release can be beneficial. And perhaps the holy grail for pharmaceutical science is delivering what we call zero-order release. That is a constant rate of release over a prolonged period of time. And this is really beneficial because what it means is you can maintain the drug concentration in the plasma at a constant level, meaning you get therapeutic benefits with no unpleasant side effects for a long period of time. And I show you this because using the electrospinning approach to make polymer nanofibers, we can deliver all of these different types of release profile. And I'm just going to show you some examples to try and convince you that I'm not lying to you and that what I've just told you is true. So we'll start with dissolution enhancement. And we'll just have a brief word on physical form before we talk about the medicine. So most medicines are given as solids because they're easier to handle, more stable, and so on. But there's a problem with that because in order for the drug to have a therapeutic benefit, I need it to get into solution. So I need the drug to transition from the solid state into solution. And most drugs occur as crystalline materials. That means they have a very regular arrangement of molecules in the solid state, and that means they have strong intermolecular forces between those molecules holding them together. So that can be a major problem because I need to go from the solid to the liquid to the solution state, and to do that I have to overcome these interactions between the molecules. And that requires energy, and it takes time. And it's particularly a problem now because more and more drugs are poorly soluble. So my boss, Professor Craig, who was in Colombo about a month or so ago, he has a slide he uses in some of his presentations where he puts up a picture of the Taj Mahal, and he tells the audience that a large number of drugs coming into the market now are less water soluble than the Taj Mahal. So we are literally trying to dissolve brick dust. And that's because the interactions between the molecules in the solid state are very, very strong. So I tell you all of that because it can be very beneficial to make what we call the amorphous form of the drug, where the drug molecules in the solid state are randomly arranged, because that means there are no intermolecular interactions, and it means there is no energy barrier to dissolution, and therefore we should get a very fast transition from the solid to the solution state. And because electrospinning is really fast, it's very good at making amorphous materials. So my strategy here is I take the drug, and in this particular example, we use paracetamol and caffeine, two drugs commonly given together in Europe, at least for the treatment of colds and influenza. And we spun those into fibers using this thing, polyvinyl pyrrolidone. So this is a very fast dissolving polymer in water. And so that means that by making the fibers, we have both the benefit of the drug being amorphous, but we also have this fast dissolving polymer, which is really going to help everything move into solution, or so we hope. So we made some fibers with different drug loadings, ranging from 10% to 35% paracetamol. You can see we have nanoscale fibers. They're about between 200 and 500 nanometers in size. As the drug loading goes up, the fibers get bigger. That's not a surprise because I'm just processing more material in a unit time. If we look at what they look like by eye, these are scanning electron microscope images. If we look by eye, then they look kind of like a bit of tissue paper. And we can cut them very easily, and we can shape them very easily. So we can make a nice round disc, or we can make a U shape, which is this one here, and we can use that under the tongue. We can use that sublingually. So these are designed for application directly to the mouth, for drug release in the mouth, for instance, rapid relief of symptoms in children, or for elderly people who have dysphagia where they can't swallow, or indeed for psychotics where you want a really rapid release of drug because they might spit their medicine out before they've had time to take it. If I have a tablet of paracetamol and caffeine, that will disintegrate and dissolve very slowly. So you can't see this very clearly, for which I apologize, but this is a tablet of paracetamol and caffeine which I put in some water and just left to stand for 10 minutes. You can kind of see there's some disintegration, but there's also still some solid at the bottom here. After 25 minutes and a bit of stirring, you put a cloudy suspension here. whereas the fibers dissolve much faster. And if this works, there's a nice video to show that. So what we're looking at here is this is a high-speed camera image. So this is recorded at about 1,000 frames per second. Where is the time? You can see the time here. So this is 40 milliseconds here. So what you're seeing is, in the middle of the image, I'll get rid of this stupid error message. Here is the electrospun fibers cut into a disc, and they're just being lowered into a Petri dish full of a saline solution. So just not the artificial saliva. And if we play the video, we will see that as soon as I add the fibers to the liquid, they disintegrate. So this is 200 milliseconds now. You can see as soon as they're added, they're gone. So what are we at now? 340. So within 300 milliseconds of adding the fibers to the saline, they have completely disintegrated. That means as soon as you put them in your mouth, they've disintegrated. Before you notice it, they've disintegrated already, and you've already taken the medicine. So if you were a child, or an elderly person, or a recalcitrant psychotic, then you've already taken your medicine, whether you wanted to or not. I won't show you the rest of the video, because it's quite boring. So that's disintegration. That's the solid map breaking up into small lumps. If we look at dissolution, by which I mean the rate at which the drug molecules become a solution, hence the liquid phase in solution, then F5, the black line, this is a physical mixture of paracetamol and caffeine. We just took the two powders and put them together. And this is percentage release plotted on the side. So 100% release means all the drug that was in the formulation is freed into solution. So for the physical mixture, we can see over about 6 minutes, 360 seconds, we get about 50% release of the paracetamol on the left, about 80 of the caffeine on the right. Whereas the fibers, F1 and F4, they're just formulations with different drug loadings, the details don't matter. Within 150 seconds, we have 100% release of both drugs, and particularly for paracetamol, the release rate is markedly faster with the fibers. We can also incorporate a taste masker. I don't know how much people use paracetamol in Sri Lanka. Is it commonly taken? Yeah. It tastes horrible, right? If you accidentally leave a tablet on your tongue for a second too long, it's really disgusting. And ibuprofen is worse. So we can add a taste masker, put a little bit of raspberry flavour in, and what we find is that it masks the taste very nicely. I can't show you the data because we kind of just did it in the lab. We didn't get ethical approval, so it's not in the paper if you look it up. But I promise you it works. You can add the taste masker, and all you taste is a little bit of raspberry, a little bit of sweetness. So we can do really fast release. So I can make these oral fast-dissolving drug delivery systems, which I spoke about, which we can use maybe for the treatment of children, for the treatment of old people who can't swallow, and so on. So what about delayed release? Well, we can do that too. And the way we do that is we just make fibers out of a pH-sensitive polymer, a Utrigit, something like that. So this particular example, we made core shell fibers, and they look a bit like this schematically. So the core contains polyethylene oxide. It's a grass polymer generally regarded as safe. It's also mucoadhesive. And we have some drug embedded in it. And then we made the shell out of a polymer called Utrigit S100. So Utrigit looks like this. It's a methyl methacrylate-based polymer. And these family of polymers are very clever because you vary the amount of ester and acid groups that you have, and that lets you vary the pH at which they dissolve. So this particular polymer, S100, is completely insoluble in water below pH 7 because all of these acids are protonated, so it's not charged, it's not ionized, it doesn't want to dissolve. When you go above pH 7, these things become ionized and the polymer dissolves very freely. So Utrigit S100 is widely used to target release to the kernel and places like that. Polyethylene oxide, we know. The functional component, we chose two drugs in this study. The one on the left is mebeverine chloride, the one on the right is indomethacin. And the reason we chose them is because, first of all, they have the benefit of this one is basic, this one is acidic, so we can see the effect of the drug acicity or basicity on the release. They have similar molecular weights, and also there is utility in delivering both locally to the colon, because mebeverine hydrochloride here, this acts locally on the smooth muscle in the intestine, and you need local action to relieve the symptoms of irritable bowel syndrome, which it's used to treat. Mebeverin can be used to treat colon cancer. It's not in the clinic, but there are lots of preclinical studies in the lab which suggest that it can be powerful. So there's a benefit to delivering those locally to the colon. So we made some core shell fibers, and what we're looking at here are transmission electron microscopy images. And hopefully you can see, we'll focus on the bottom left one, which is the simplest one, that in all cases, the fibers have very clear core shell morphology. There are two clear compartments. You can see here the core in dark, and the shell is a lighter color. And the same is true for all of them. The middle one here is also very obvious. The fibers aren't completely beautiful. There are some irregularities in their shapes, and that upset the student doing the work. But if you can make the same thing 3 times and get the same results, I don't think it particularly matters what they look like, although the students like pretty fibers. So if we look at the drug release from these, then I should have used grey and white. That was a schoolboy error, wasn't it? But hopefully what you can just about make out is the first 2 hours of this release experiment are done at pH 1.2. That's to mimic the stomach. And then we take the fibers out and we transfer them to a pH 7.4 medium, which is mimicking the small intestine. So the MV formulations at the top, they're the Mebeverin formulations. The 2 graphs are just made of different molecular weights of polyethylene oxide. And in both cases, what we see is in the first 2 hours, there is a bit of drug release, but certainly less than 20%. At the bottom, we have the indomethacin formulations, and there is no release at all there in the first 2 hours, literally nothing. So what this is telling me is, if I put the drug in these core shell fibers, I can prevent release in the stomach, in the case of the acidic indomethacin. And if you look in the literature, when people do this sort of thing, they almost always use acidic drugs, which is cheating, right? Because the acidic drug is in any case going to be much less soluble in the stomach than the basic drug. So we weren't particularly surprised by this, but we were quite pleased by these results. You do get some release of the mebeverin at low pH, but not much. In most cases, less than 10%, which fits with the US Pharmacopoeia requirements for a delayed release formulation. I was going to say something else about that, and I forgot what it was, so I won't say it. So by making these core shell fibers, we can prevent release in the stomach, and therefore we can target release to lower parts of the intestinal tract. We can also use this sort of approach to make what we call theranostic materials. So materials which contain both an active ingredient and also something to allow us to image it. So if, for instance, I'm treating a tumour in the colon, it would be really nice if I could monitor how big the tumour was during the treatment period to see if the medicine's having an effect. So we made some fibers, there they are, SCM images and TIIKM images, some core shell fibers, basically the same recipe as before. So the shell on the outside is Utrojic S100. In the core, we have polyethylene oxide, and here we have some indomethacin, and this thing, gadolinium DTPA. Gadolinium DTPA is used for magnetic resonance imaging. It's a contrast agent. And if we look at the release profiles, we see basically the same thing as in the previous slide. Very little release at pH 1, sustained release over between 8 and 24 hours at pH 7.4. So we can make more complex materials. We can make theranostic materials as well. Okay, so fast dissolving, delayed release, we can do that. What about pulsatile release? Turns out we can do that too. So here what we did is we polyethylene oxide, our friend again. It's very good for electrospinning. But this time we blended it with sodium alginate, so naturally occurring polysaccharide, and it also has some pH sensitive properties. So depending on the balance of these different units, then it's going to be soluble below above roundabout pH 5, something like that. And sodium ibuprofen is our model drug here. So we took these 3 and we blended them into fibers. And I haven't said this explicitly, but normally what we get when we do the electrospinning is the fibers collect as a flat mat, just like a piece of paper. In this case, we don't see that at all. We see these 3-dimensional mountain structures that are growing upwards from the collector plate towards the needle. And we don't really know why that is, but we believe it's because there are lots of charge carriers present. So in the previous examples, pretty much all the materials we're spinning have been charge neutral. Whereas here, we have sodium alginate, sodium ibuprofen. We had a lot of charge carriers. And actually what we find with this experiment is if you touch the needle, you do get a really nasty shock because there's a lot of things building up the charge there. But if we look at the electron microscopy images, we can see we have fibers. So these are SEM images taken from the bottom, these two, and from the top, these two, showing the fibers. And this is STEM, just looking at the internal structure of the fibers. You don't see much. They look pretty much monolithic, the fibers. So we can make the fibers. And we made a bunch of formulations with different amounts of polyethylene oxide, alginate, and ibuprofen. And at pH 6.8, we see this. So everything releases in 15 minutes. So the student did this experiment, and then he was really disappointed. He was really bored, because those are uninteresting data. I've made a very complicated immediate release formulation that has no benefits at all, right? But then he did the experiment at pH 3. And to this day, I'm not really clear why, because it's not a standard pharmacopoeia pH to look at. But it is representative of the stomach in the fed state, and also often in elderly people who tend to have reductions in stomach acid production as they age. And what we see at pH 3 is we see very nice pulsatile release. So pretty much as soon as the formulations are added to the pH 3 medium, they release some of the drug. Then there's a lag period where nothing further is released, and then there's a second burst. And you can see these are the ibuprofen contents here. So F3, which is the low ibuprofen one, is releasing about 10% of its drug loading in the first burst. F2 about 40%, F3 is around about 60-65%. So there's nice correlation between the amount of ibuprofen present and the amount of release in the first stage. And so these formulations could be very useful for pulsatile release if they remain in the stomach, and we can also use them as like dual trigger release systems almost. So over about 2 hours, you would expect that the formulation will be transferred into the small intestine. So if that happens, we get a burst in the stomach, then we get nothing, and then we get another burst at pH 6.8. But if you've had a big dinner, or just through random variation, sometimes it does take longer for the drug to exit the stomach. And so if no further release happened in the stomach, that's potentially a problem. But happily, we have this second burst of release. So if you do have a delayed gastric transit, then the drug will be released anyway. So there are benefits through that. So we can get this nice pulsatile release, and we can tune the amount of release in the first stage by varying the amount of ibuprofen present. We can also vary the lag time between the two phases by varying the amount of alginate. So these are further formulations made going from green with 6.3 up to orange with 14.3% sodium alginate. And you can see, as we go across here, the lag time between the first burst and the second burst gets longer. The more alginate there is, the longer you delay. And in fact, with this orange formulation, the student told me he ran the experiment for 4 days and he still didn't see the second burst of release starting. So nothing is going to, well, I really hope that nothing's going to stay in your gastric transit for 4 days. If it is, you've got a serious problem. So this orange formulation is completely useless for anything, but very interesting that we can tune the amounts of release and the lag between the release just by varying the components, or rather the proportions of the components in the system. So we can do pulsatile release too. We can also, if we are clever enough, we can do zero order release. So zero order release is generally really very difficult to achieve using standard tablet and capsule formulations. There are huge amounts of work going on trying to achieve it. To do zero order release, we have to get a bit cleverer. So we've done single fluid electrospinning so far, 1 needle, 1 liquid, and we've done coaxial, 2 needles, 2 liquids. Here we went even more complicated and we did triaxial. So we just have 3 needles, inner, middle and outer, and in each one you have a different fluid. So your needle looks a bit like this. And this looks incredibly simple, absolute nightmare to do, takes ages to optimize, but it can be done. Here's a picture of the experiment. So you can see that we just put some dyes in the fluid to help us visualize what's going on. They don't do anything, just tiny amounts of them present. But they do you can see the outer fluid is this green colour, the middle is pink, and the inner is blue. And so that means that if you look at the cone that forms, remember I told you that the droplet deforms into a cone. If you look at the cone, then you can see the outside appears colourless, then there's a pink layer, and then there's a blue layer. So we can see that in this cone we have 3 layers. And here is the needle here. 3 needles, one nested inside another. So what we did is we made the fibres, and all 3 layers are made of the same polymer. So this is unlike the Udrid examples where the outer layer was Udrid, the inner layer was polyethylene oxide, which I can't say. In this case, all 3 layers are made of this polymer, ethyl cellulose, which is water insoluble, and they all contain the same drug, ketoprofen, some non-steroidal anti-inflammatory. But they contain the drugs in different amounts. So the numbers here are really important, but in the outer layer we have about 4% drug, in the middle layer it's about 17-18%, and in the inner layer 40%. So as we go from the outside of the fibres to the middle, we see that the amount of drug as a percent weight is increasing. So the first thing we did is we just spanned those 3 fluids on their own. So just 3 fluids, ethyl cellulose and ketoprofen dissolved in, I think, ethanol if memory serves, with different drug loadings. You can see from scanning electron microscopy, they look the same. And if we measure the fibre diameters, again, they look about the same. The more drug there is, they get a bit bigger, but nothing exciting is happening here. If we then spin the 3 fluids together in the triaxial spinneret, then again in the SEM, here we can see nice fibres. The size is about the same as it was spinning the fluids individually, and you can see the fibres look monolithic. But if we look at them by transmission electron microscopy, this image never comes out really very well on the screen, but I promise you it's true. You can see that the outer layer is very light in colour, and then there is a darker grey layer, and the middle layer is completely black. If I put some lines on the image, okay, that's clearer. So what we've made is we've made a 3-layer fibre. All the layers are made of the same polymer, they contain the same drug, but the weight composition is different. And the drug content increases going from the outside to the inside. Okay, so if we then look at drug release from the monolithic fibres, the fibres where I spin each of the 3 ketoprofen ethyl cellulose solutions on their own, then what I see is a plot like this. So you can see the percentage release increases with time, reaching about 80%. All 3 different solutions behave about the same. What we also see is the initial release rate is much faster than the release rate at the end of the experiment. And that's completely standard for electrospun fibres. We almost always see a burst of release at the start of the experiment, which then tails off at the end. And the reason for that is the fibres have really massive surface area to volume ratios. So that means that a lot of the drug is at the outside of the fibre, and it doesn't have far to diffuse into solution at the start of the experiment. Whereas the drug in the middle has to go a long way to get out. So this shape of plot is very typical. And it's exactly the same shape of plot that I've shown you in the previous examples, except that then it didn't really matter. Because when I'm trying to do a very fast dissolving thing, I want it to come out very quickly as possible. When I'm doing delayed release, what I want is a lag time, nothing for a bit, and then ideally some fast release. And with the pulsatile release, this is bursts of on-off release. And I want bursts, rapid acceleration, then nothing. But if I want to sustain the plasma concentration of a drug at a particular level for a long period of time, then that burst of release is potentially going to be a problem, because I'll see a very rapid initial increase in the plasma concentration, but then as this rate tails off, the plasma concentration will drop, and probably it will go below the minimum effective concentration in the bloodstream. So what we're trying to do is to get away from this burst of release and then this tailing off. And since I've told you all that, you won't be surprised to know that we can indeed do that with these 3-layer fibres. So this on the right is the same plot but with the triaxial 3-layer fibers we have. And you can see the release rate is linear between 4 and about 20 hours. We start to get a little bit of tailing off at the end, but we have a very nice linear fit here. So we have 0 order release between 0 and 20 hours. So what that means is you can wake up in the morning, you can take a tablet of your medicine, and then you can sit back happy in the knowledge it's releasing its drug at a constant rate in your body throughout the day. Long-term relief of symptoms, no side effects. So happy days. So how does all this work? I don't want to bore you with the details, but I will a little bit. So these are the fibers after the experiment. We recovered the fibers from the dissolution experiment, and we looked at them under the electron microscope. And so you can see they're kind of shriveled, they're wrinkled on the outside, and there are lots of gaps in the middle, lots of pores in the middle where all the drug has been released. So the fiber is insoluble. The polymer here is basically completely insoluble in water. So that means in order to get out, the drug has to diffuse through the polymer into solution. And if I have a monolithic fiber, a fiber which is the same all the way throughout, then there are two effects. At the outside, there is very high surface area, there is lots of drug. It doesn't have far to go to get into solution. So it will diffuse out very quickly. But as we go from the outside to the inside, the surface area is decreasing, and the drug has to diffuse further out to exit the fiber. So that's why we see these plots where rapid burst of release and then a tailing off. With the 0 order fibers, with the 3-layer fibers, what we're doing is we're compensating for the increased diffusion distance by having a greater drug concentration. So the drug in the outer layer in grey doesn't have very far to go to get into solution, so it comes out very quickly. And the drug in the light blue layer, the middle layer, that has further to go, so normally we would expect it to come out more slowly, to give us a slower change in concentration per unit time. But because we've increased the drug concentration there, we've balanced out this effect. So although the drug has further to go, there is more drug in the middle layer, and that means that the rate of release in terms of milligrams per second or whatever is the same, or almost the same. And the same thing is true again on going to the inner layer. The distance to diffuse is further, but the concentration is increased again. So the gradient distribution of drug helps to compensate for the distances in diffusion, the differences in diffusion distance, too many D words there, and allows us to deliver this 0 order release profile. So I'm not going to pretend that a 0 order release profile of ketoprofen is particularly useful. I mean, it's a low value drug, and similarly pulsatile release of ibuprofen is probably not that useful either. But the point is that these are proof of concept studies to show that we can deliver these types of release patterns using electrospun fibers. So these are complicated dosage forms. That means that they're going to be expensive to produce, particularly if you have to check that your batches of fibers are all 3-layer, are all giving 0 order release. And that means they are most applicable to high value products. So very potent actives, new actives, maybe actives which have very particular delivery requirements. But by using this approach, we can produce all these different delivery systems. And all of these materials were made on that same set of apparatus you saw at the start of the experiment. One set of apparatus can do all of these different things by the simple expedient of changing the needle I use and changing the polymer that I make the fibers out of. So I wanted to finish by talking about something a little bit different, by talking about some tissue engineering. But before I do that, something very important, I think, is to talk about the industrial applicability of these fibers we've seen a little bit. So all these fibers were made in my lab or in my collaborator's lab, and our rate of production is, we're probably processing about 2 mils of solution per hour at a polymer concentration of maybe 10% weight volume in solution. So we're producing about 200 mgs of fibers per hour. That is not very good, unless you are trying to make something extremely high value and very potent. So we have some other apparatus which lets us scale up to about 80, 100 mils an hour. So we can start to produce maybe 8 grams of material an hour. which is better. It's enough for pilot studies and things, but it's still not going to produce enough material to treat the population of Sri Lanka or indeed any other country. But in the last 5-10 years, there have been really massive developments in scale-up electrospinning. And we have some collaborators in the lab in Hungary, and they have equipment which can process hundreds of litres of material per hour. So hundreds of litres times about 10, they can probably produce, they're producing on the kilograms per hour scale, which is not unreasonable for industrial production. And so they showed me some calculations they did, and they found that, and I forget what medicine it was exactly, I think it was a prednisolone formulation, they could make within 1 day enough material to treat the population of Hungary for a year. They also found they can take the fibres and they can onward process them. So the formulations are very fast dissolving formulations. We probably don't want to just use those as oral films. So we'll want to make a massive sheet, and then we'll want to cut them to shape, and then we package them and give them to the patient. The fast dissolving polymers are going to be hygroscopic, so that means we're going to want to seal them under vacuum or package them under nitrogen, which adds a little bit of cost, but is not prohibitive. I mean, crisps are packaged under nitrogen to keep them fresh, and if you can afford that for a bag of crisps that costs, I don't know, 100 rupees, you can most definitely afford to do it for a medicine. For the other formulations, probably we want to take them orally, and so the easiest way to do that is as a tablet, because it's just a bit harder to swallow a lump of tissue. And you can cut the fibres, you can process them into tablets, so that's absolutely not a problem. It doesn't have an effect on the properties. So, tissue engineering is, I guess, increasingly exciting, and people are applying increasing amounts of attention to look at it, so I'm not going to talk about it very much. But we've been working on this project for the last few years, which I think is quite nice and which I wanted to share with you. And what we're trying to do is we're trying to make a bioresorbable replacement for the right ventricle outflow tract. So we'll unpack all of those things one by one. The right ventricle outflow tract is a conduit in the heart, and it's a conduit in the heart that very frequently goes wrong when children are born with heart defects. And I forget the numbers, but lots of children die of it every year. So if you're lucky enough to live in a country with good hospitals, then what will happen is when the parents notice that there's a problem, or the doctors notice there's a problem, the baby will go to hospital and they'll be fitted with a replacement for the faulty right ventricle outflow tract, or RVOT for short, RVOT. And that's great, except the surgeon will put in a bit of plastic to replace the defective part of the heart. So that's all right, but children grow, hopefully, and plastic will not grow. So what will happen is the child will get older, it will get larger, its heart will get larger, and the conduits in the heart, the tubes in the heart, will also get larger, except the artificial one. So when the child is maybe 4 or 5, the surgeon will have to go in again, open them up, do another operation, but in a larger replacement, and again in a few years' time. And this causes a lot of problems. Operations are deeply unpleasant. There is always a risk of death or other side effects, however skilled the surgeon may be. And they're also enormously expensive for healthcare providers. So if we could get away from that, that's money freed up which could be spent on other things, other new medicine. So there's an increasing move to develop what are called bioresorbable implants. And those are implants which degrade in the body over a period of time to safe metabolites, basically. So what we're trying to do is we're trying to make a scaffold which goes in the body temporarily, which gets overgrown by the body's own cells, and as it gets overgrown, which dissolves and is degraded away. So it's a temporary replacement, a sacrificial template to help regrow the body's own tissue where it was defective originally. So this is a project funded by the European Union, and it involves lots of partners in lots of different countries. So that's us. We also work with a lot of clinicians in a hospital, and we work with some companies who do medical polymers, I guess polymers for medical applications, a research institute in Germany, and this company, Celiat, who are the commercialization people. They're supposed to be the ones who take it to clinic. So the first thing I have to do is, if I'm going to make a replacement tube, I need to make my fibers into a tube. So I could just take a flat mat, roll it up and sew it together,
but that's not very satisfactory. There'd be weaknesses in the stitches. So what we want to do instead is to spin a tube. So this is how we do that. Rather than collecting the fibers on a flat mat, they're collected on this cylinder, which rotates at high speed. It's going at about 6,000 RPM, I think, or something like that. And here's my needle with my polymer solution being ejected from it, and the needle moves up and down the tube in order to build up the scaffold shape. How long is this video? A bit boring, let's skip ahead a little bit. So after 5 hours or so, we have begun to build up a tube. You can see there's a shadow here. That's my postdoc, Bilal, who's about to remove the tube. There he goes. Guest appearance by his beard on the right-hand side there. And so here we've built up a three-dimensional tube. This is one of our early prototypes, so it's a bit ugly. I'm not allowed to show you the later ones for intellectual property reasons, but they're better, I promise you. So we made a tube. That's nice. Looks a bit like this when we get the good one. So when we're making these types of scaffolds, then the scaffold really has to do two things. The first one is that it has to replicate the functions of the body's own tissue in the short term. And so if you're in the heart, there's a lot of blood flow, there's a lot of shear and stress, so it has to have suitable mechanical properties to recapitulate, to replace the functions of the native tube during that time. So our partners, Vescal, did a whole bunch of mechanical tests, which I'm not going to talk about in detail, but we measured the modulus, the tensile strength, all those sorts of things. And I always tell my students, never put tables in presentations, but I also tell them, do as I say, not as I do, so I'm going to allow myself to have a table. The numbers aren't important. Really what's important is to notice that they change a lot. So these are tubes made of different materials, and I can vary the modulus from 26 to 1 megapascal, for instance. So by changing the polymer I use, also by changing how fast I rotate, how fast I dispense liquid, I can vary the mechanical properties. And my aim is to vary those so that they match the natural properties of the heart. We also want to look at the biocompatibility in vitro and in vivo. So here are some tube samples. These are controls without cells. And then these are experiments to grow stem cells on them. The purple colour just because the cells have been stained. And you can see that cells grow on them very nicely. You see a very nice layer of cells on top of the mat. You're starting to get some cell penetration into the fibre mat. And that's important because we want the cells to penetrate inside in order to replace the beta structure. And we also need to look at biocompatibility in vivo. So these experiments are done by our collaborators in France who are very skilled surgeons. What they do is they take a mouse and they give it a heart attack. That's a pretty hard thing to do. So you open the mouse up, you tie basically a bit of string around one of its arteries for a little while to give it a heart attack. Then you untie the string, sew it up, wait for a bit, check it's had a heart attack. Then you open it up again, and what you do is you sew in a little tiny patch of our electrospun fibers to try and replicate, to try and recapitulate, help the heart recover from the trauma it's undergone. So this is after I think about 6 weeks, and it's really hard to see. So here's the way the patch has been sutured on. And in fact, it's really hard to see is good, because the body's own tissue is colonizing it, is replacing the patch. In a rat over 6 weeks, in a person we want longer, probably a couple of years, more time to play. And all of these things I'm going to talk about so they really understand them, but basically all of these are telling me that the cellular growth over the implant is normal. This is what it should look like. You see the normal cell populations, they show all the normal surface markers and so on that I would expect. So at the moment, well, I might have died, but the last thing I heard, our latest prototype is currently inside a lamb. It's been, so this is a complete tube now. It's been in a lamb for, I think, around about 6 weeks, and at the time of speaking, unless something bad happened over the weekend and no one's told me yet, the lamb is still alive, which is excellent. Because we can't eat it when it dies, it's apparently against the ethics, which is very sad. So, by making these sorts of electrospun scaffolds, because the fibers kind of mimic the structure of the extracellular matrix, you get very nice cell growth of them, you tend to get very nice cell penetration, and if you can design them to have the right mechanical properties, then you can use them really effectively in order to make bioresorbable implants. And there are some other guys doing something similar. They're a bit further ahead of us, a company called Exceltis, and they have, I think, 6 of their materials inside children now. I think it's Uzbekistan. They did the trial somewhere a bit crazy because they don't have many ethical rules over there. But as far as I'm aware, the children are all alive and well. And so these approaches really do have the potential to make a major difference to people's lives. And that, ladies and gentlemen, is really all I want to say. I hope I've convinced you that I could, at least as the first one, I can produce drug-loaded polymer fibers. There's loads of evidence for that. And if we choose the polymer, and if I choose the structure of my fiber cleverly, then I can make materials that give me a range of drug delivery profiles. Delayed release, pulsatile release, very fast release, and so on. And we can use these materials in tissue engineering as well. And therefore, in my view, electrospinning is a cool technique, and it's a technique which is increasingly scalable, and therefore it's also an applicable technique. There are some electrospun products on the market at the moment, but they're mainly for filters and things like that. So when people remove asbestos from buildings, for instance, you could get masks that are made of electrospun fibers to stop you inhaling the particulates. So I should finish with some acknowledgements, because I did absolutely none of the experimental work myself. It was all done by these people. So Iranka Lanka, Sri Lankan guy, graduate from Colombo University, a special program in pharmacy. He did all the fast dissolving drug delivery systems. Yassine did the pulsatile release. And Miao Dong did the delayed release systems. And my collaborator Ben Wong Yu at University of Shanghai for Science and Technology is the mastermind behind the zero-order release systems. And loads of people helped us with teaching. And I'd also like to thank the Royal Society, the National Science Foundation of China, and the European Union for money, without which we couldn't have done any of it. And I will finish by thanking you for listening.